Hello, my name is Brian Lee, currently in ninth grade, and today I'm going to be going over my analysis of human brain samples with Alzheimer's disease versus healthy control samples. To begin, what is Alzheimer's disease? Alzheimer's disease is a progressive neurological disorder that primarily affects the brain, leading to memory loss, cognitive decline, and changes in behavior. It's the most common cause of dementia, a term used to describe a range of symptoms related to decline in memory and thinking skills. Alzheimer's disease is characterized by the buildup of abnormal protein structures in the brain, which disrupt the normal functioning of nerve cells and eventually lead to their death. Currently, there is no cure for Alzheimer's disease, but treatment and interventions can help manage symptoms and regulations. My slide, sorry. My hypothesis is that there will be a significant difference in protein levels between individuals with Alzheimer's disease and healthy controls. As we can see in this photo, sorry, I don't know how to remove this. As we can see in this photo, we can see a difference between a healthy brain compared to those who suffer from Alzheimer's. We can see, we can see a huge shrinking in the cerebral cortex and hippocampus, along with enlarged ventricles. We can see that on the neurons, there are neurofibrillary tangles and flakes. This photo also shows physical effects of Alzheimer's and its severity. To analyze this protein level data set, I utilized the GEO2R tool, commonly used for analyzing various types of data, including protein structures. For the analysis, I labeled the samples into two groups: Alzheimer's for samples obtained from individuals with Alzheimer's disease, and control for samples obtained from healthy individuals, based on the experimental conditions and metadata provided in the dataset. The specific GEO's dataset number used for this analysis is GSE 138260. After labeling the samples accordingly, I performed statistical analysis using GEO to identify proteins that are differentially expressed between the Alzheimer's and control groups. This process involved comparing protein expression levels across the two to identify proteins that are significantly more or less abundant in Alzheimer's disease compared to healthy controls. This volcano graph goes over its relevance and abundance to its effect on people who have Alzheimer's, while the mean graph collects the average brain activity for each protein across both groups. The box plot graph represents any signs of outliers that are in the dataset. The volcano graph y-axis indicates that the higher value stronger evidence against the hypothesis, while the x-axis shows that the common representation for changes in protein levels between conditions and experiments. I gathered two underexpressed proteins and two overexpressed proteins. The first underexpressed protein is a COX1, which is an enzyme that is involved in the synthesis of a prostaglandin, which is a lipid molecule in the role of inflammation regulation. The second one is the ND2. The connection between NADA subunit and Alzheimer's disease lies in their association with mitochondrial dysfunction, oxidative stress, and neurological damage, all of which are key features of Alzheimer's pathology. The overexpressed protein consists of cytochrome C. When changes occur in the cytochrome, it can contribute to mitochondrial dysfunction, ultimately leading to neurodegeneration and cognitive decline. The second protein is the ATP synthase, which relates to Alzheimer's due to its role in cellular energy production and mitochondrial function. The box plot graph was used to show its consistency throughout the data. As the boxes are all similar in shape and position along the y-axis, we can understand that there are no outliers in this data set. My conclusion is that there is a significant difference in proteins between individuals with and without Alzheimer's. Understanding this significance can provide the next steps toward a deeper understanding of this disease. Thanks for watching.